Imagine this. A disease that, well, barely harms its host, causes really minimal symptoms, almost never kills.
Correct.
Yet, it can shut down entire national economies, cost billions. Sounds like a riddle, doesn't it?
It really does. What are we talking about?
Today, we're diving deep into the fascinating and honestly pretty paradoxical world of swine vesicular disease, SVD for short.
SVD, right.
And to help us unpack this, we've waded through a lot of material, comprehensive veterinary reviews, epidemiological studies, even insights from vet exam prep materials.
Good stuff, the essentials.
Our mission today is really to tackle that core paradox. Why does this relatively mild pig illness trigger such huge, costly, globally disruptive responses?
Yeah, why the massive overreaction, seemingly?
Exactly. We're going to unravel the surprising reasons behind its, let's say, outsized impact. Let's get into it.
Let's do it.
So swine vesicular disease, SVD, what is it fundamentally?
Fundamentally, it's a contagious viral disease, and crucially, it only affects pigs, domestic pigs, wild pigs.
Right. Not cattle or sheep.
Nope, just pigs. It's caused by the Swine Vesicular Disease virus, SVDV, which is an enterovirus.
And the symptoms, what does it look like?
The classic sign is these fluid-filled blisters. Vesicles is the technical term.
Vesicles, okay.
Yeah.
Where do they usually pop up?
Primarily on the feet, especially around the coronary bands. That's sort of where the hoof meets the leg skin.
Gotcha.
You might sometimes see them on the snout or the lips, the tongue, but less often, really.
But here's the twist you mentioned earlier.
Yeah, and this is critical. The infections are very often really mild,
Yeah.
sometimes even subclinical.
Meaning no visible signs at all.
Exactly. The pig looks fine. And mortality. Death from SVD, it's negligible, practically zero.
So the direct hit to the pig's health or farm productivity,
Yeah.
it's minimal.
Tiny, almost insignificant in itself.
Okay, so why all the fuss? Why
Yeah.
the global importance?
Right. The profound global importance, the economic chaos, it stems entirely from one fact. Its clinical signs are indistinguishable from foot and mouth disease, FMD.
Ah, okay. And FMD, that's the big one, right?
That's the nightmare scenario. FMD is, well, it's probably one of the most economically devastating animal diseases on the planet. Transboundary, spreads like wildfire.
And it affects more than just pigs.
Oh, yeah. FMD hits all cloven-hoofed animals. So cattle, sheep, goats, wild ruminants, plus pigs. High morbidity lots get sick, spreads incredibly rapidly, sometimes even through the air.
Wow.
And in young pigs, FMD virus can cause this acute heart issue, myocarditis, they call it tiger heart, leads to sudden death. It's catastrophic.
So a vet walks onto a farm, sees blisters on a pig's feet. They can't tell if it's the mild SVD or the catastrophic FMD just by looking.
Exactly. They look identical in pigs, and that's the core problem.
Which leads to
Yeah.
what? What happens immediately?
It leads to the guilty until proven innocent principle. International regulations are absolutely clear on this. Any vesicular disease in pigs
Any blisters.
must be treated as a potential FMD outbreak until the lab proves otherwise, period.
And the consequences of assuming it's FMD.
Immediate, massive, and expensive. You're talking instant quarantines, halting all animal movement in the area, intensive surveillance. It throws everything into lockdown.
So the economic damage isn't really from sick pigs.
Not directly, no. The real economic hit comes from the trade restrictions. Disease-free countries immediately slam the door shut, embargoes on pork, live animals, you name it.
Right.
Those indirect costs, plus the cost of eradication programs, which usually means culling, the stamping out policy for infected herds in any contacts.
Stamping out sounds drastic.
It is. Humane culling of potentially thousands of animals. The costs are just staggering. They dwarf any losses from the actual SVD illness itself.
Okay, so the mimicry is key.
Yeah.
But you mentioned the virus itself has some interesting features. Its origin.
Yeah, this is fascinating. The SVD virus, SVDV,
Yeah.
it's actually a pig-adapted version of a human virus.
Wait, the human virus?
Yep. Specifically, Coxsackie virus B5, or CVB5. Genetic work strongly suggests a single jump from humans to pigs.
A cross-species jump from us to them.
Exactly. Scientists call it zooanthropognosis, and they estimate this happened sometime between, say, 1945 and 1965.
And the first SVD outbreak in pigs was?
1966, right after that estimated jump. The timing is pretty suggestive.
Wow, okay. And what family does this virus belong to?
It's a picornavirus in the Picornaviridae family. Same family as FMD virus, actually. These are generally known for being pretty tough little viruses.
And you can track it.
Yeah, that's another interesting bit. While there's only one main serotype, like one main flavor, there are at least 4 distinct genetic variants that have evolved over time.
So you can follow its spread.
Precisely. Molecular epidemiology lets scientists trace outbreak pathways almost like genetic fingerprinting. It's really powerful for figuring out how it moved.
But what makes it so incredibly hard to control on the ground?
I'd say the single most defining biological feature of SVDV is its just extraordinary environmental resilience. This thing is a survivor.
How resilient are we talking?
We'll think about pH. It's stable from pH 2.5, that's highly acidic, all the way up to pH 12.0, which is very alkaline.
That's a huge range.
It is. And for a virus without an envelope, that fatty outer layer that often protects viruses but can also make them easier to kill with disinfectants, it's relatively heat resistant too. Needs like 60 degrees Celsius for 30 minutes to reliably kill it.
So cold doesn't bother it much either.
Not at all. Refrigeration, freezing, preserves it just fine. And all this stability means it lasts for weeks, even months, in the environment.
Where? Like in the soil?
In soil, yeah. And manure slurry, on contaminated surfaces. This is why fomites are such a huge deal for SVD.
Fomites?
Yeah.
Inanimate objects, right?
Exactly. Contaminated trucks, farm equipment, boots, clothing. These become major ways the virus spreads because it just sits there waiting.
You can't just get rid of the sick pigs and be done.
Absolutely not. The environment stays contaminated. It's a nightmare for disinfection.
And you mentioned something about food products earlier.
Right. This is really concerning, too. The virus can stay infectious in processed pork, even after things like curing, salting, smoking, fermentation.
Seriously, like in salami or ham.
Yeah. It's been found to survive in processed sausage casings for over 2 years, in dried salami for over a year, in cured hams up to 180 days.
6 months in a cured ham. That's mind-boggling.
It tells you just how incredibly tough this virus is and why it's such a headache for international trade.
So this extreme hardiness, this resilience, that's really the key factor driving the control strategies.
Absolutely. It dictates everything. Simple culling isn't enough. The environment is a reservoir. You need incredibly harsh disinfectants, rigorous cleaning, and often long fallow periods where farms have to stay empty before restocking.
It really highlights the virus's survival skills.
Definitely.
Okay, so it's tough. How does it actually get into a pig and start spreading, especially if it's often so mild?
The main ways in are ingestion eating contaminated stuff. The classic example is uncooked garbage or swill that contains infected pork scraps.
The swill feeding issue.
Exactly. Also, direct entry through breaks in the skin or mucous membranes, little cuts or abrasions, which are pretty common in pigs housed commercially.
And once it's in?
It starts replicating locally, like in the tonsils. Then it hits the bloodstream pretty quickly. That's called viremia. You can often detect the virus in the blood within 24 hours, sometimes with a fever.
And the viremia spreads it around the body.
Right. It seeds secondary sites, especially those epithelial cells in the feet, snout, mouth. And that's where you get the characteristic vesicles, the blisters.
But here's the scary part, right? The timing.
Yeah, this is a huge challenge for control. Infected pigs can start shedding the virus, releasing it into the environment up to 48 hours before they show any signs of being sick.
So perfectly healthy-looking pigs can be spreading it.
Precisely. They're infectious before anyone knows there's a problem.
And how is it shed? Where does the virus come out?
Multiple routes. Nose, mouth secretions, urine, semen, definitely the fluid from burst vesicles. But the most significant route, especially for long-term environmental contamination.
Let me guess. Feces.
Bingo. Fecal shedding is huge, and it can go on for a long time, up to 3 months after the pig seems to have recovered clinically, often in really high concentrations.
3 months of shedding virus in manure.
Yeah. Imagine the buildup in the environment.
And this ties back into the mild or invisible infections.
Absolutely. That's the other critical epidemiological piece, the high frequency of mild and subclinical cases. In many outbreaks, maybe even most of the infected animals show no obvious signs.
But they're still infected.
They still get viremia, they develop antibodies, and crucially, they shed infectious virus in their feces. They become silent reservoirs and spreaders.
Making detection incredibly difficult.
Exceptionally difficult. It's like trying to control something you often can't even see.
So let's put ourselves in the vet's shoes again. They see blisters. It's an emergency. Because it could be SVD.
Or FMD. Or vesicular stomatitis, VS, or Seneca virus A, SVA, or even vesicular exanthema of swine, VES, though that's more historical now.
5 different diseases that look the same in pigs.
Clinically indistinguishable in pigs, yes, all causing vesicles.
And because FMD is the potential catastrophe.
Rapid, definitive lab diagnosis is absolutely non-negotiable. You must assume it's FMD until proven otherwise.
So in the field, before the lab results,
Yeah.
Are there any clues, anything to help differentiate?
The best tools you have right then are good detective work, epidemiology, and looking at which species are affected.
Okay, how does that help?
Well, take vesicular stomatitis, VS. The big clue there is if horses are also showing blisters, along with maybe cattle or pigs.
Horses get VS.
Yes, and cattle and pigs. VS is also spread by insects, and its zoonotic humans can get a mild flu-like illness from it.
Interesting. What about FMD?
If you see blisters in cattle or sheep or goats, in addition to pigs on the same farm or in the same area, the presumptive diagnosis has to be FMD.
Got it. What about Senecavirus A, SVA?
SVA mostly hits pigs. It's emerged more recently globally since around 2014. It can look just like SVD, but it's also linked to something called epidemic transient neonatal losses,
Meaning?
meaning sudden high death rates up to 50% or more in very young piglets less than a week old. That's a potential clue for SVA.
And VES, the last one.
Vesicular exanthema of swine. Really only historical for the U.S., linked to feeding uncooked marine mammal scraps or fish. Declared eradicated from U.S. domestic pigs back in 1959, so less likely now, but still on the list of possibilities.
But even if it's only pigs showing signs, you still act as if it's FMD.
You absolutely have to. Until the lab results come back negative for FMD, the emergency FMD protocols stay in place. The risk is just too high.
So lab tests are everything. What kinds of tests are we talking about?
Broadly, two types. Tests to find the virus itself, the agent, and tests to find the body's response, the antibodies.
Okay, finding the virus.
You've got rapid tests like antigen detection ELISA, good for high virus loads like in blister fluid. But the real workhorses, the most sensitive and specific, are PCR-based tests. RT-PCR, real-time RT-PCR.
Detecting the virus's RNA, its genetic material.
Exactly. These are crucial, especially for finding those subclinical infections, maybe from fecal samples. And the really clever thing now is multiplex RT-PCR. Yeah, one test that can simultaneously look for SVD virus, FMD virus, VS virus, maybe SVA virus, all in the same sample. Incredibly efficient for differentiation.
Wow, that's huge.
It is. And then there's virus isolation, actually growing the virus in cell culture in the lab. That's still considered the gold standard for absolute confirmation that infectious virus is present.
And the other side, detecting antibodies.
That's serology, looking for the immune response. Antibody detection ELISAs are great for screening lots of serum samples quickly, the backbone of surveillance programs.
And confirming those results.
The virus neutralization test, or VN test, that's the international gold standard for confirming antibody results, making sure they're specific and ruling out false positives.
So given the subclinical problem, just waiting for reports of sick pigs isn't enough for surveillance?
No way. Passive surveillance, just relying on clinical signs being reported, is totally inadequate for SVD. You need active surveillance, meaning going out and systematically collecting samples from apparently healthy pig populations and testing them, especially using things like pooled fecal samples with RT-PCR to find those silent infections.
Okay. Now, despite all these challenges, the mimicry, the toughness, the silent spread, you said SVD eradication has actually been a success story in many places.
Largely, yes. It first popped up in Italy in '66, then spread through parts of Europe and Asia in the '70s and '80s.
But then it was pushed back.
Yeah, through really concerted national and international efforts, most countries managed to eradicate it by the mid-'80s, primarily using that tough stamping out policy.
Which involves
Rapid detection, strict quarantine, humane culling of infected and contact animals, followed by incredibly thorough cleaning and disinfection.
Italy was a tougher case, though.
Italy was the real long-term challenge. The virus became endemic, really established there for decades. It took a massive, sustained effort.
What did they do?
A comprehensive program, super strict movement controls, high biosecurity standards, mandatory disinfection protocols, and just enormous active surveillance, testing huge numbers of animals.
And it worked.
Eventually, yes. Italy was officially declared SVD-free in 2015.
That's quite a turnaround.
It really is. And Italy's success story kind of demonstrates that even with a really difficult virus like SVDV, incredibly resilient, spreads silently, eradication is possible.
What does it take?
Sustained political will, serious investment in robust surveillance, good biosecurity, and international cooperation.
Looking back at how it spread, what were the main drivers?
Consistently, number one is the movement of those subclinically infected pigs, animals that look healthy but are shedding virus.
Right.
Number two, contaminated fomites, especially transport vehicles. That environmental stability of the virus makes trucks a huge risk if not cleaned properly.
And number three?
Still, the feeding of uncooked garbage, swill, containing infected pork scraps, a classic route for many pig diseases.
So boiling this down to prevention, biosecurity is paramount.
Absolutely. It's your first line of defense. Strict rules about where you get your pigs from, quarantine for new arrivals, minimum 30 days, controlling who and what comes onto the farm people, equipment, shower in, shower out protocols, farm-specific clothing,
And vehicle cleaning.
rigorous vehicle management, thorough cleaning and disinfection before a truck comes onto the premises.
Yeah.
And the disinfection part for SVDV has to be serious.
Because it's so tough.
Exactly. Your standard disinfectants might not cut it. Often you need strong alkalis, like sodium hydroxide solution. If the cleaning and disinfection isn't done right, the virus can just come right back. It's a common reason for outbreaks recurring.
And at a national level.
Strict import controls on live pigs, semen, pork products, and either banning swill feeding or having very strict rules requiring proper heat treatment to kill any potential virus.
And if an outbreak does happen,
SVD is a notifiable disease worldwide. Suspicion means immediate reporting. That triggers official action. Quarantine, movement bans, setting up control zones, usually a 3-kilometer protection zone right around the infected farm, and a wider 10-kilometer surveillance zone.
and the stamping out policy.
Yes, that typically follows. Humane culling of all pigs on the infected farm and potentially others that had contact. It's harsh, but it's proven effective for eradication.
One thing we haven't talked about is vaccines. Is there an SVD vaccine?
No, there are no commercial SVD vaccines available or used.
Why not, if it causes economic chaos?
Two main reasons, really. First, the disease in the pig itself is generally just too mild. It doesn't cause enough production loss or animal welfare issues on its own to make routine vaccination worthwhile from that perspective.
Okay, and the second reason?
This is the bigger one, especially for trade. Vaccination creates antibodies, and the antibody response from a vaccine would look identical in many standard lab tests to the response from a real infection.
So you couldn't tell if a pig had been vaccinated or actually infected.
Exactly. It completely messes up surveillance. You lose your ability to prove your country is actually free of the disease, which is crucial for exporting pork and pigs, so no vaccines are used.
Makes sense from a trade perspective. Okay, final area.
Yeah.
What about people?
Yeah.
You said the virus originated from a human one. Can it jump back? Is SVD a risk to public health?
That's a really important question. Despite its origins in that human Coxsackie virus B5, the risk of SVDV transmitting from pigs back to humans is considered negligible. It's not seen as a significant public health concern.
So no cases in people working closely with infected pigs.
There are no documented cases of farmers, vets, slaughterhouse workers getting SVDV from occupational exposure during outbreaks. The only confirmed human cases ever were a handful of accidental infections in labs.
Laboratory accidents, what happened?
They were generally mild flu-like illnesses. One case was associated with meningitis but recovered. These were isolated incidents in a lab setting, not out in the field.
So for the general public?
Risk is essentially zero. For people working with pigs, it's considered negligible. And for lab workers, it's managed through standard biosafety protocols. It's really an animal health and economic issue, not a public health one.
Okay, so let's wrap this up. We started with this paradox. A mild disease causing massive disruption.
Right. SVD is unique, defined not by what it does to the pig, but by its uncanny resemblance to the truly devastating foot and mouth disease.
And its global significance, the billions in costs, it all stems from that mimicry, forcing these aggressive control measures primarily to protect international trade.
Exactly. And layered on top of that are the virus's own tricky characteristics, that incredible environmental toughness.
Surviving for months.
Its surprising human ancestor and its ability to be shed for ages by pigs that look perfectly healthy, all making it super challenging to find and eliminate.
Yet the eradication from most of the world is a major success story, a testament to veterinary services and cooperation.
It absolutely is, but the threat isn't gone, is it?
No, the potential for reintroduction is always there. Or maybe even new viruses emerging from similar human enteroviruses jumping to pigs again. Vigilance is key.
And we're seeing other FMD mimics pop up too, like Seneca virus A more recently. It just reinforces how critical robust surveillance and really tight biosecurity are across the board.
Definitely.
So maybe a final thought to leave everyone with. What does this whole story of swine vesicular disease really teach us? How can these seemingly small biological details, a virus looking like another one, its ability to thrive outside a host, have such enormous, far-reaching consequences for global trade, for food security, even for our daily lives? It makes you wonder, How much of our animal disease control is really about protecting the animal, and how much is driven purely by economics?